Dr. Deneyer's background is a PhD in molecular and cell biology from the University of Louvain in Belgium. In 2019, she began work for GSK, where she coordinated scientific projects aiming to deploy next-generation sequencing for quality purposes. In January 2021, she joined the GSK Global Quality Control Department as an NGS quality expert. Her topic today will address the emerging ability to detect replication-capable virus in serum. Her title for today's presentation is NGS Transcriptome coupled with metabolic labeling as readout of infectivity assays to specifically detect live viruses in serum. Please welcome Dr. Deneyer.
Good afternoon or good evening, everyone. My name is Deneyer, and I'm working as a scientific expert at GSK Vaccines Belgium. And today it's a pleasure to be here to present you how next-generation sequencing, or NGS, can be used as a readout of infectivity assays to specifically detect live viruses in raw materials such as serum. So this work was sponsored and funded by GSK, and the work was outcontracted to Charles Rivers and PathoQuest, which is a spin-off of the Pasteur Institute specialized in NGS. So the objective of my presentation today is to propose you a new perspective on how NGS could be used in industry to control the risk of viral contamination in vaccines, but also in raw materials such as serum. And NGS could be used here not only as a detection assay, but also a mitigation tool in the context of the viral safety. Because yes, this work was performed under a viral safety context. And as I'm sure you know, viral safety is really critical for vaccine manufacturer. And for each product, we have to establish a risk assessment. And for the risk assessment, we have to take into consideration three main pillars, which are the starting materials. So depending on the cell bank, on the viral seed used, it will impact the risk assessment. Also, we are taking into consideration the potential GMP failure during the manufacturing operation. And finally, we are taking into consideration the animal-derived raw materials and the potential contamination that could occur from these raw materials. So interestingly, recently, it has been published that raw materials were determined to be the most likely source of contamination in biological manufacture. And this is for us a really key point to be considered today. So, but what is happening and how we are building a risk assessment? So first, we have to assess the risk. We have to identify the potential viruses that can be present based on the three pillars. We have to analyze and evaluate the risk. And at this stage, we are taking into consideration the viral knowledge and the diversity that we know thanks to publicly available data. Based on this risk assessment, we have a list of viruses of concern, and we have another concern here is that we have an increasing risk due to new viruses and new variants that are not known for the moment, for instance. Based on this list of viruses, we have to provide a risk control strategy through risk acceptance or risk reduction. And for risk reduction, we have different kinds of strategy to propose, such as in the case of a raw material, the selection of the raw materials, viral clearance studies, such as irradiation of a serum, for instance, and also detection assays, such as the 9CFR assay for serum. However, while this kind of assay presents a large advantages, there are more and more limitations that are highlighted by regulatory agencies in the context of this kind of infectivity assays. For instance, only cytopathic viruses are targeted by CPA detection. So we are not targeting non-cytopathic viruses. Also, depending on the readout of this kind of assay, new variants and known viruses are maybe difficult to be analyzed. Also, it's sometimes difficult to discriminate between inert nucleic acid present in a sample and live viruses, and this can lead to false positive results. And so recently, new assays such as PCR assays were proposed to overcome some of these limitations. Indeed, it's possible through PCR to analyze non-cytopathic viruses, because you are targeting through the PCR a specific sequence of the virus, and you have to see whether this sequence is present in your sample or not. So this is okay for non-cytopathic viruses, but this kind of assay does not overcome other limitations, because you have to know the sequence that you are targeting. So new variants and unknown viruses, it's difficult. And also with PCR, you are targeting all the nucleic acid present. And so if a virus was present before the irradiation of your serum, for instance, it will be positive with a PCR, while this sequence is maybe not coming from a replicative virus and will not induce a viral safety risk. So this is why today we are proposing other type of assays such as NGS, because as PCR it will also analyze non-cytopathic viruses, and in addition it will also covers new variant and unknown viruses, because this technology is not based on previously known sequence. So it can detect everything in the samples, even if we don't know the sequence of viruses, for instance. But, and this is the goal of the presentation today, we still have to demonstrate that NGS can make the distinction between inert nucleic acid and live viruses, to not be in the same scenario as PCR and to have false positive results. So this is the goal of this presentation today, and as a teaser I can already tell you that it's working, and NGS can be used to discriminate between inert and live viruses. So what is NGS? So NGS can be really summarized easily, as you can see here, through a different steps, and starting from sample generation, such as, for example, your cells after the infectivity assays. Then you have a step of RNA extraction. So you will have to extract the different RNA from your samples. Then you have to prepare your sample for the sequencing it, and then after you have a really important step, which is the bioinformatics step. Indeed, this bioinfo will provide different answers to the question raised at the beginning. And so, a short example: if in your sample you had 3 different viruses and you don't know which kind of viruses it is, well, at the end of your NGS, you can have, through bioinformatical assays, you can identify, for example, known viruses, a variant of a known viruses, but also an unknown viruses. So this is really the strength of NGS. So in our context of viral safety, here we would like to show how to use NGS for our specific question, which is how to detect living viruses in serum. Well, if a virus is present in a serum, once you are putting this serum on cells, the viruses begins to replicate in these cells, and so the viruses through this replication will generate and will produce viral transcripts, so viral RNA, and this RNA can be seen as a marker of virus replication in infected cells. And so, thanks to the sequencing, we will follow 2 types of biological responses. The first one is an increase of the number of viral sequence reads. Indeed, along the cell culture period, you will have an increase of viruses, and so an increase of viral RNA that will be produced in the cells. And this can be followed by NGS. At least we will show today that it's possible to use NGS to follow that. Also, another biological response that can be followed is the detection of newly synthesized RNA through the addition of a metabolic labeling. And this will also be covered by this presentation today. So thanks to NGS, we will be able to follow these two kinds of biological responses that will be linked, that are linked to viral replication. And so we will have the possibility to follow replication signature in cells. So to answer the need and to show that NGS can be used for that perspective, we designed a proof of concept assays, and for it we selected 4 kinds of viruses to really cover the panel of viruses that can be found in the nature. So we selected the human adenoviruses 5, which is a DNA viruses non-enveloped. We also selected the parainfluenza 3 virus as well as the Coxsackie virus. We also selected bovine viruses, which is known to be non-cytopathic and which cannot grow on MRC5 cells. So what we have done is that we spiked these 3 viruses in FBS, and then we cultivate these infected serum on MRC5s for up to 21 days. We did the same for the BVDV viruses, which were also spiked on serum and culture on MDBK bovine cells for up to 21 days. So different samples were collected after 0, 7, 14, and 21 days. And also in parallel, we observed potential CP, and we also performed hemadsorption and hemagglutination assays. So now we are really entering into the result part, and here is result for the first biological response. So if you remember the slide with the cells, you can see that we can follow the replication of viruses in cells through the increase of viral sequence in our samples. And this is what I would like to show you now. So for instance, for human adenovirus, we spiked 1 TCID50 in the serum, and we cultured it for up to 21 days. And at day 0, you can see that in the samples, we observed only 9 specific sequence of human adeno in the samples. Why there is some adeno? It's because for the day 0, we infected the cells with a really high dose of viruses for 2 hours at 4 degrees to mimic non-replicative viruses. So this background can be linked to a virus that bounds the cell membrane. So it's normal here to find background for these specific viruses. So at D0 there is 9 specific reads, while at D21 there is more than 100,000 reads for the human adeno. So it's really clear. You can really see here a clear increase of specific human adeno during the cell culture. So we did the same for human PEV3, and also here you can see a really strong increase between D0 and D3 this time. For Coxsackie, also the same. You have here the numbers at D0, at D7, and D11. And what is really interesting in our case is that even at D7, you can have a really nice and clear increase of the sequence identified by NGS, while the CPE was negative at this period. So it seems that NGS can identify the viral sequence before a positive CPE signal. And finally, here is the result for the BVDV virus. And again, you can see here a nice and a clear, strong signal after 21 days. So to conclude, you can see here that the NGS can really identify a net increase of viral reads for all the 4 viruses in scope of this study, and even for Coxsackie, NGS had the possibility to detect reads before CPE signal. But, and this is something that can have an importance for daily use, this kind of biological response and the use of NGS for it requires more than one time point, because you have to compare between D0 and D3, 7, 21 days. You have to really have these 2 time points to compare and to see an increase of number hits, and this can be a limitation in daily use. So just maybe a short parenthesis is that thanks to the bioinformatical analysis, you can also have, and you have to do deeper bioinformatical analysis to exclude background and to specifically identify replicative virus. And one of these analyses is the coverage, the percentage of coverage. Indeed, if you have just background and if you have, I don't know, a risk that can be found elsewhere and which is not specific to viruses, you will not have full coverage of the virus genome. And to show you that, here you can see the genome of the human adeno5 virus, which is on the axis of this graph. And here you can see the number of reads across this genome. And as you can see here, we have 96% of coverage. So it means that we can find reads over nearly all the entire genome of adeno. And this is really a point to consider, because now we know that we have replicative viruses thanks to the result I just showed you, but this is logical also in terms of virus presence, because you don't have only reads, I don't know, localized in a really short fragment of the genome. So here the reads are covering all the genome, which is important for this kind of assays. So for the Coxsackie, the PEV3 or the BVDV, we have a percentage of coverage between 81 and 100%, and this is really important to consider in our case. But let's go back to our biological responses, and let's now go to the second one, which is the use of metabolic labeling to identify newly synthesized RNA. So for that, the principle is quite easy. So if you have a viruses in the serum, once you have you put it in a culture on cells, as already explained, the virus will replicate and will generate RNA, viral RNA as marker of replication. And what you can do at the end of a cell culture is to apply metabolic labeling, which will incorporate into newly synthesized RNA. So meaning the RNA that are synthesized only during the treatment period. And so you will have two different kinds of viral RNA. RNA that is transcribed during this treatment, so the newly synthesized RNA which incorporated this labeling, and RNA that were previously there. For an example, it can be RNA present before an inactivation of the serum, for instance. And so what you can do, thanks to the bioinformatical analysis, is to identify RNA where this labeling was incorporated, because this will lead to a specific mutation in the RNA. And so this mutation is observed here in blue. And so you can have the distinction between here RNA where the labeling was not incorporated because the RNA was there previously. And so you can see here that you have a background noise in terms of mutation, but it's kind of logical. And then here you have the RNA which incorporates the metabolic labeling, and you can see here that this mutation is really increasing. And so you can see the distinction between inert viruses and live viruses. So these are the results that we obtained for PE3, but we also did it for the three other viruses. So with adeno, you can see here clearly the distinction of the inert viruses with live viruses through this mutation rate. For PEV3, you already saw the results. We did also it for Coxsackie, of course. So here we have one condition in our experiment what was missing, but based to other samples that were not present today, we are confident that this is also working for Coxsackie. And finally, for BVDV, you can also see this net increase of mutations in the condition where we had the labeling and which corresponds to live viruses. So this is a summary of the differentiation, conversion, and mutation index that we observed in our experiment. So as you can see, we have a mutation rate which is a background for all the conditions except the one where we put the labeling and where we spiked, of course, live viruses at the beginning. So this is really interesting because it means that through this NGS coupled with metabolic labeling, we can specifically detect newly synthesized RNA. And another point to consider here is that we also measure this mutation for the cellular genes, for MRC5 and MADK genes, because once you apply the treatment, it will also incorporate in the transcript of the cells. And this is really interesting because it can be used as an internal control to be sure that the treatment was correctly applied on the cells. However, for what it's really working for MRC5 genes, we have to still optimize it for MDBK genes. So as a conclusion, today I show you that the NGS assay represents a promising tool and method to detect and identify virus sequence in biological samples, and then before CPE detection. We show also today that we can really discriminate infectious viruses from inert, and that the replication can be applied normally to all types of viruses. So I showed you two methods today. First one without the metabolic labeling. So if you remember the one where you can see the increase of a specific viral rate in time. And so this is really a nice way to do, but it requires more than one time point because you have to compare between two time points. And then I show you also another method which apply metabolic labeling to specifically detect newly synthesized RNA. And this is really interesting because you only need one single time point and culture condition to have the results. So in terms of perspective, so to use this approach, it still requires and it will benefit of future optimization, such as you have seen, an optimization for bovine cells internal control, so using the bovine cellular genes, but this is in progress. And as a take-home message, I hope that today you learned that the NGS transcriptome approach coupled with metabolic labeling as a readout of infectivity assay could be used to confirm the absence of live viruses in raw materials used for vaccine reproductions, including serum, and to show that the viral sequence are not corresponding to live viruses. So this is really a detection tool as well as a mitigation tool. And for us, we believe that this approach could be used to test raw materials as a guarantee of high level of viral quality. And we hope that these assays will be used for raw materials, including serum, of course. So I would like here to thank all my JSC colleagues which were involved in this study, as well as PathoQuest members and Charles Rivers for performing this different work. So thank you for your attention, and please feel free to ask questions.